we just want a physical thing. Yeah, that's the word that shows. Yeah.
Can I turn this over?
Sure.
So we can do
It's up in my office. We'll get it after class, okay? I'm going to leave that there. Happy Friday. I just had lunch at Woody's. I didn't have an hour to prepare for class, so I'm going to make it up today. So, I'm going to talk to you about directing mutations. We're going to talk about mutations. I'm going to tell you how humans direct mutations into organisms. And then we'll talk about how you can use those directed mutations to make a genetically engineered organism. And then I'll tell you a little story that's going to blow your mind. And then I'm going to tell you another story that's going to blow your mind. You guys ready to have your mind blown? Fasten your seatbelt. So when I say homologous recombination, does anyone know what that means when I say homologous recombination?
Is it recombination of homologous chromosomes?
Recombination of homologous chromosomes, when does that happen?
In meiosis.
Meiosis? Crossing over in prophase 1, right? So that's another way, homologous recombination. So we have machines that we've learned about that line chromosomes up and then physically cause the DNA to exchange over, right? So what we could do is we could have a chromosome with some gene on it. Let's say we have the bad copy of cystic fibrosis on this, okay? So this person has cystic fibrosis. It's in every cell in their body. Pardon me. Woody's Pizza is killing me, I guess.
Oh, it's so good.
It was so good, but it's killing me now.
What's your
And then-
favorite? Let's talk about pizza.
Let's talk about pizza instead of blowing your
Pizza
mind.
beer.
I talked about beer this morning in vodka. I'll talk about, no, we won't talk about pizza. Nice try. So if someone, let's say this person has inherited two bad copies, two mutant copies of the cystic fibrosis gene, so that it's a chloride transporter, it's not functional, their lungs fill up with all the goop, and they're probably going to die when they're in their 20s. So there are treatments that we can do to potentially prolong that person's life. But the challenge is that person is going to, if that person has children, the person will pass those defective alleles on to their progeny, right? So can we go into the genome and modify the genome and put a wild-type copy in there, at least in the gametes, like in a sperm or an egg, so the progeny of this person will have a wild-type copy. Make sense? So the way to do this is to clone the normal version of the gene, just like you were in lab. Now you are actually going for cDNA sequences. What you need is the genomic version of it, right? With all the introns and exons and everything in there. But the same idea is the same. Is you go to a genome that has a normal wild-type copy of that. And you find restriction enzymes on either side, and you cut the DNA and isolate this. You run it out on the gel. And you know what size this will be because we have the human genome and we know where this will be cut. And then literally you take that, you take a plasmid and you cut the plasmid with the same enzymes, and now this can be ligated into the plasmid. Now probably the genomic structure of this is so large that the plasmid is so small by comparison. It probably goes like this. Okay. Now I have specifically chosen restriction enzymes that have sequence outside of the gene, right? There's sequence here and sequence here, so I call this flanking sequence. Okay? Now, I can take this plasmid, yes?
Is that not unstable?
In general, it's fairly unstable. If you put this in bacteria, bacteria are not too happy with it to grow it up into large amounts, but there are tricks that people have learned, and they learned those as a result of the Human Genome Project.
Okay.
So let's say you get in a lab where this is standard operating procedures. So we can take a cell out of this person, and we can have that cell growing in a dish. With the 2 mutant chromosomes in it. And we can take this plasmid and through various tricks get it into the cell. Like this. And this sequence matches this sequence right here, right? So this sequence here matches that sequence right there. And we can even, when we put the plasmid in, we can even put some proteins in. And the proteins are the machine that does homologous recombination. So what we do is, maybe we'll have a plate where we're growing up, and we have 100,000 cells in here. We treat 100,000 cells like this. In some of those cells, this plasmid will recombine such that this clicks off of the plasmid into the genome. And the mutant copy exchanges and goes now onto the plasmid. Does that make sense? So we have caused a chromosome to recombine, to cross over with a plasmid. And this is standard. It's not hard to do anymore. When I was in grad school, it was hard to do. It's not hard to do anymore.
It's not hard to do it in a petri dish, not in a person.
Yeah, it's not hard to do it. Yeah, so but we do it in a petri dish.
Yeah.
Okay. And then, so now this person is no longer homozygous recessive. They're a carrier. And you could probably repeat the process and try to get another cell where the other copy gets recombined out, right? And there will be a lot of PCR that's needed to determine if you've got the sequences swapped out. We won't get into those details, but in a dish, you can actually make this. It might take you 6 months. Because I'm terrible in lab, I'm going to give myself 2 years, okay? All right? So now we have a cell from that person growing in a dish. If we could take that cell and somehow get that to be the germ cell that is the source of all the gametes, now that person we would have corrected this mutation. Does that make sense? So we can actually do things like this in mice. So let's say we want mice that we want to modify a biochemical pathway so that the mice are really efficient at that, so that they burn fat really easily. Something like that. We can modify the genes in a cell in a dish like this. And then we can do something really incredible, because mice don't complain about ethics when we do things like this. Right? So we can take an egg, which is 1N, and we take the nucleus, we take an incredibly small needle, and we stick that in here, and we suck the nucleus out of the cell. And now have a ghost of an egg. It's an anucleate, or akaryotic, a non-karyon egg. Does that make sense? I can then go to one of these cells and take the nucleus out of it, which has this genome, and I can take that needle and stick it in here and inject that nucleus into this egg. So the egg was 1N, it went to 0N, but now it's 2N. And what happens is the egg goes, oh crap, apparently I was fertilized. I didn't even know. And it will divide and divide and divide and grow into an organism based on this DNA. It's karyotic transfer. They're transferring the karyon transfer, the nuclear transfer. sometimes it can be tricky to get this to grow into a mouse. It doesn't grow right. So we have another trick. You're going to love this. I'm going to leave that up just because I like it. So we can actually we can take an egg, drop a sperm on it in the mouth for the mouse, and have that divide. And if you guys remember from, I don't think we teach it in our intro bio courses here. The cell divides and divides and divides, and you wind up with something that looks like this. This is a sphere that's hollow. There's liquid in here, but these are all cells that are genetically identical. This is called a blastula, and it's a fertilized egg that is divided a whole bunch of times to make this thing. This, for simplicity's sake, this is going to become the placenta, and this mass up here is going to become the organism. Okay? What we can do is we can take one of these cells that we've genetically modified, and we take a needle and we stick that cell into this mass. And then we take this and implant that in a female mouse, and it implants in the uterus and grows into an organism, such that the organism is this is a mouse. The mouse had whiskers. And we're going to say that that cell was injected into the spot that developed into this ear of the mouse. We have a mosaic organism. An organism which is mostly this, but one of the ears is genetically different than the rest of the organism. Make sense? So researchers who are wanting to make genetically engineered mice will take 100 of these, and they'll inject a cell into each one of them. And every now and then, the mouse develops such that the genetically modified cell grows into this part of the mouse. So that the gonads are genetically different than the rest of the organism. So that all of the sperm produced by the organism are genetically this and not genetically this. What a dirty trick to these cells, huh? So if this organism mates, it will pass on these genetics instead of its own genetics. This is standard stuff. Okay. I talk about this in a number of my classes. One time, a couple decades ago, there was a ship that the ice had just melted north of Russia. And the first ship going through that oceanway north of Russia there was going along, and someone on board was on deck and looked over, and the ice, a bit of the ice shelf had broken away and fallen down, and laying on top of the ice was a frozen baby elephant. And they're like, what the hell is a baby elephant doing way out here in the Arctic area? So they landed at the next little village, at which point someone said, I don't think that was an elephant. I think that was a baby mastodon that got frozen at the start of the last ice age. And the ice age is still winding down such that the ice just melted and that thing just became exposed. And so that is a perfectly preserved baby mastodon that is 14,000 years old. And so they raced back in their boat, and the scavengers, like wolves and bears and such, had not yet found it. So they gathered up the body and they froze it back down, and it has been preserved ever since. Could you take the nucleus? Is the DNA preserved? That you could take the nucleus out of one of the mastodon cells and put it into an elephant egg and implant that into an elephant, and what would grow would be a mastodon.
Probably not.
Probably not. Why? Yeah. Is the DNA intact?
Maybe bits, and then, just those are stuff that just happens.
So probably the DNA is there, but not fully intact. So it might be that you take the nucleus and you add a bunch of DNA repair proteins into the nucleus
That might help.
to ligate the DNAs back together.
But even then, you're still probably not going to get perfect.
Okay.
But there's progress.
The question is, do you want to go to the zoo someday and see a...
I do.
Yeah.
So,
There's a whole series of movies on why that's a bad idea. Jurassic Park, yes. But we will be much smarter than that.
don't bring back the scary.
Yeah, don't bring back the scary things, right?
No.
Bring back the things that
We're just
are cute and fuzzy and mammals. Right?
no one's there
Okay.
Did they try to do something like that at some point?
To the best of my knowledge, they have not tried to do it.
I thought I read something about the mastodon trying to recreate it or whatever.
They would like to.
There's really not much to work with, so if you mess it up, sucks.
Yeah.
We have another 1,000 years for another one to pop out, so nice one.
So there are people now that scour these Arctic areas looking for things that were frozen like this.
Now is the time to see if the ice is.
Yep. Okay. You ready for the really good one? That was good, wasn't it? You ready for the really good one? All right. I want you to explain to me how this. I'll give you the data. You tell me how it came to be. There is a woman who gave birth to a child, and there were people who watched the child come out of the woman, right? At some period of time, she needed assistance from the state or something from the federal government in like welfare type stuff. And they needed a genetic test. You know, maybe she kidnapped some kid or something, right? They want to make sure if it's biologically your kid, you can get the assistance or something like this. When they did the test, she is definitely not genetically related, or she is not the mother of the child. She's not a surrogate. She didn't have some other woman take eggs that were implanted in her. Without a doubt, the child there came out of her. There's no question about it. They can show. There's no mix-up at the hospital where, whoa, this kid got the wrong tag and the wrong kids went home. She's a relative of this person, but not the mother.
Can't support there,
Can anyone guess as to how that happened? What happened?
She a genetic mosaic.
She's a genetic mosaic. How did that happen?
Eternal twin.
A twin. So her mother's womb, her egg came down. The egg that was this woman came down, got fertilized, and implanted. And a different egg also simultaneously came down, got fertilized, and implanted. And they implanted right next to each other, such that they grew together to make one blastula. And the most of her body was her, but her ovaries were her sister. right? So it would be an aunt. She'd be the aunt of her own kid. Genetically?
My aunt actually kind of had a similar thing, but her twin was more like a tumor to her.
Oh,
So she had to get it removed.
Wow. When did they figure out it was a tumor?
When she was like a teenager.
Can you say where it was?
I don't know.
Wow.
Was her twin still? Wait.
It was a tumor. It was, instead of like it being her ovaries, it was just a tumor.
That's what happened to my cousin too.
Wow.
And the tumor was in her ovaries.
The tumor was in her ovaries. So they had to go in and take that out. Wow.
Did this happen to like male sperm?
Yes.
Spermies?
Yes. So there are 40 documented cases of this. 40 documented cases of this.
That's just documented.
Yeah, and that's just documented.
Doing this stuff pretty recently.
Yeah.
How odd.
How odd. Did that blow your mind?
That's pretty cool.
But the twin was never like born, right?
Sure.
Like
I mean, they were both born simultaneously.
so they were. It wasn't like
They were in
They
one
were
body.
one person.
Yeah, they are one person. It's just equal.
what?
Genetic
That's
makeup.
what's so weak. That's okay.
Yeah. Anyway. All right, I think that's what I've got for today. I'm running out of steam. That pizza, I had too much of it. Should we get out of here early or no?
I need lunch so bad.
Yeah. You can help us.
Oh yeah.
Help you with the problem?
Yeah.
Okay, leave that running. You guys need help with this problem?
Yes.
Okay.
Alice. Yeah. If you want to.
So this is how I did it. I put every possible genotype on there.
Usually it's like my twin.
Yeah, that was pretty deep, but it was yeah, I just like fell over and just like myself.
I hate him.
Right? What are the other possible combinations?
All little.
Okay, what else?
All dominant, all wrong.
Is all dominant a possibility? Yeah, it is. I guess it'd be this. Okay?
Oh, those people obviously are, right? Call them up. Look us up.
Right? Is that right? I think those are all the possible combinations, right? So I think the odds of this coming up were 50%. Right? And the odds of that coming up were 3 out of 4. Right? And the odds of that was another 50%. Yes? So the odds that someone would be born with this genotype is 1 over 4, 3 over 16. Is that like 18% or something like that?
18.75.
What?
18.75.
18.75. Right? So you could do that for each one of these. You calculate each one of these out, right? But I think this one doesn't count. This one doesn't worry. We don't need to worry about that one because we're going for deafness, right? That person was normal hearing. So all of these are the deaf phenotypes, right? You guys understand how I did that? How I came to this? You look like you don't understand.
Which one is not? The one is all recessive is not deaf? I thought.
No, this one.
Oh yeah, okay. Sorry, I missed
That one's not deaf. So that's not part of the question. I mean, the easy way to solve it is to figure out the odds of that and subtract that from 100. That's the chances of all of these other possibilities, right? Alright, so I said, what are all of the possible genotypes that could lead to deafness? So I picked this one, and there's an 18.75% chance. And I think this one was 18.75% also. But this one was not. This was less than that. 6%, something like that. Right? So I figured all of these guys out. And then you add them all up. And I think, what did it come to, like 85% or something?
Yeah. Close to it.
Somewhere around there. So that's part one. And the reason it was so easy then for part two, you know, you're reading, you're like, oh, 50% are penetrant unless this or that, or oh, what the hell are you talking about, right? But the way it actually works out is, if something has if a person is homozygous recessive at just one trait, the bones are actually formed just enough that about half the people can hear, half the people can't hear. So what that means, if it's 50% penetrant, then instead of 18.75%, it's 9% of those guys are deaf, and 9% can actually hear, even though they ought to be deaf. And the same thing here, and the same thing here. This is 9%, this is 3%, because this was what, 6% or something like that?
And then all the other ones are the same because they got the double dose.
Yeah, but these guys are deaf because of this and this, and the way the question was worded, I said, if people are like this, the number doesn't change. This is 100% penetrant. This one, and this one, and this one, and then this is 3. All 3 are recessive. Does that make sense? So these numbers don't change. All the only change are those numbers, and so this goes down to like 60% or something like that. So once you set it up for part 1, part 2 was easy, I thought.
In your mind, we didn't think about it that way, I guess.
So when I designed it, I was like, wow, this is an awesome question. Because I'm giving them the amazing opportunity to do this 1, 2, 3, 4, 5, 6, 7 times. It's 7 questions in one.
Oh boy.
And then I test their understanding of penetrance and the terminology of homozygous recessive or at 1 trait or 2 traits or whatever. I give them the opportunity to shine on all those concepts too, all in this one question. This is a great question. And then I went to answer it to make my answer key. I was like, oh man, this is really hard. It took me a while to do it. And I had to do a spreadsheet because I was cheating. Well, I did it by hand, and I was like, I don't trust my answers.
How do you like spreadsheet? Like AA, or did you just like spreadsheet the fractions? Like what were you?
Yeah, I did these as text as a column, and then I said 50% for this, 75% for this, 50% for this. Well, this times this times this. This equals that times that times that. And then for the penetrance, I just said this cell equals that divided by 2. It's 50% penetrant. Or times 0.50%. Does that make sense?
Yeah,
That's how I did it.
I appreciate the explanation.
Everyone sign this?
No.
She is.
Hannah didn't sign it.
Okay.
Have a good weekend.
You too.